All right, so we are going to continue today with our discussion of digestion and absorption. All right, so reminder, our overall goals here are to understand ultimately how we're going to absorb carbohydrates, proteins, and lipids. And to understand how we're going to absorb them, we need to know how we're breaking them down, just how we're digesting them. So last time we started out with the carbohydrates. So we talked briefly about carbohydrate digestion and absorption. I'll point out something that we didn't really go into, which is how you break down lactose. So lactose is a disaccharide, and we can't absorb disaccharides directly. We need to absorb monosaccharides. So I've got this enzyme lactase. So lactase is a brush border enzyme. So if you're lactose intolerant, this is what's not working. So remembering what our digestive system looks like, I want you to let me know where you would find a brush border enzyme that could potentially break down lactose. Okay, take another couple seconds. Let's take a look. Okay, cool. Yeah, so what we're practicing here partially is just understanding terminology. So when these things are described, you can create a mental image, even if you haven't learned about that specific thing. So remember that a cell has a basolateral membrane touching the blood and kind of along the sides. The apical membrane is the part that touches the lumen, in this case, of our GI tract, right? And that apical membrane, the part touching the hollow of the GI tract, is termed a brush border because of the presence of these microvilli, which is what we're drawing when we're drawing these like loopy projections. So when we say that lactase is a brush border enzyme, what we mean is that we've got this enzyme embedded here on the apical membrane that would be touching the food as it's going past, right? So when we talked about carbohydrate digestion, we really talked about starch, which is a really large molecule, or maybe glycogen. And we saw that starch can be broken down by our pancreatic and salivary amylases. So that was specific to starch mainly. So it breaks it down into limit dextrins, which were like the corners, and some disaccharides. Okay. But we know that while we only talked about breaking down to that level, only monosaccharides can actually get absorbed into the cell. So this is an example of how that system would kind of work. So you'd have your lactose coming in, right? So lactose is a disaccharide, right? It would hit the enzyme here, right? That lactase would split into two. And then we've got a monosaccharide that we can pull in. So this is analogous to how it would work for different types of sugars as well. It's also analogous to what's going on with our proteins, right? So we talked about carbs briefly. We talked about proteins a little bit more last week, right? So we know when we eat a protein, nothing really in our saliva is breaking that down. Where we start to digest the proteins is really in our stomach. What we're doing is we know that we're secreting pepsinogen from the chief cells. We know that we have hydrochloric acid coming from our parietal cells. That hydrochloric acid in general denatures stuff in our stomach. But when we're thinking about proteins, really critical thing is that it's taking that pepsinogen, unfolding it a little bit so that it turns into this active form pepsin, which is what starts the breakdown of proteins. So we're starting this breakdown of proteins in the stomach, primarily because of this activated molecule, pepsinogen activated into pepsin, can break into smaller peptide fragments, our polypeptide chain from our unfolded protein. So here we have an example of an endopeptidase. Okay. So we did mention that we have kind of three groupings for our proteases. Proteases are things that can break down protein, right? So the ase ending is for the enzyme. That prot part is just protein, right? So as we move into the duodenum, so into the small intestine, we're going to see a bunch of pathways. We briefly looked at an image here. And one of the things that happens is that an enzyme called trypsin activates some other proteases, kind of in the same way that the hydrochloric acid activated the pepsinogen. Okay? So we have a word that describes a protease that's in a granule in its inactive form. So we're going to practice that terminology. What we would call that? Bless you. Okay, take another second and just get the three choices. And let's take a look. Okay, yes. A zymogen is an inactive form of a protease. So we often see this ogen ending on our zymogens. So like pepsin ogen was the inactive form of pepsin. We see that similar kind of naming convention as we move into the small intestine. So what trypsin would be activating when it's activating a protease, it would be slightly breaking down some zymogen in order to activate it, so to convert it into its active form. So that's what we were looking at partially when we were here in our protein digestion in the small intestine, right? So we know chyme is coming out of the stomach through the pyloric sphincter into the duodenum. So we've got chyme, that smoothie of hydrochloric acid and partially digested food, okay? We've broken down the protein a little bit with pepsin, right? So we activated pepsinogen, started to break it into chunks with pepsin. Now that we're in the small intestine, we're adding a bunch of proteases coming from the pancreas, right? So pancreas is making tons and tons of enzymes, right? It makes lots of proteins. It's doing hard work, okay? So it's coming in here through the sphincter of Oddi into our duodenum. Okay, and so here we see trypsinogen getting activated by a brush border protease, right? So this enterokinase is working in the same way that lactase would, right? So that's an analogy you could have made to understand what this brush border enzyme is doing. So here we see trypsinogen getting activated into trypsin. Okay. And we see it looping back to activate some more proteases. So here we see chymotrypsinogen, right? This ogen ending. So we have another inactive protease, another zymogen coming in. So chymotrypsinogen becomes chymotrypsin when it's activated. Here we have procarboxypeptidase. Doesn't have that ogen ending, but a pro prefix on a word does a similar kind of thing. So think like it's promoting this. It's a precursor to carboxypeptidase. Right, so this is another thing that we're activating. Okay. We have other brush border proteases like aminopeptidase, right? So creating these active proteases, so these enzymes that can break down proteins. And so we're continuing that digestive process, right? So if we have some chunks of protein, right? So some shorter bits of polypeptide coming in to the duodenum, we now have more enzymes that can continue that breakdown. Endopeptidases breaking internal bonds, exopeptidases pulling off individual little amino acids from the ends. Okay. So we talked about how that absorption works as well. So I did a little drawing before, but here's a figure from our book, right? So when we're absorbing an amino acid, we're going to cross that apical membrane by linking things up to sodium reabsorption. So it's similar to what we've talked about before with glucose, basically, right? So you can imagine this absorption happening in a similar way, right? We create a sodium gradient where we have relatively high sodium outside the cell, relatively low sodium inside the cell. So sodium likes to flow down that gradient and it can pull things along with it. So this is how we would get individual amino acids into our cells, just like we got glucose into our cells before. And then just like glucose, we'd be able to cross the other side of the cell, right? So the basolateral membrane, and continue via facilitated diffusion, because now we have a higher concentration of the amino acid in the cell. So we've got a favorable gradient moving to the opposite side. Okay. Dipeptides and tripeptides are a little bit bigger. So when we're thinking of carbohydrate, we can only absorb monosaccharides. That's not exactly true for proteins. For proteins, you could get a dipeptide or a tripeptide into the cell across that apical membrane. We have mechanisms for doing that, but you'd need to break it down before you get it into the blood. So in that case, we'd have additional enzymes inside the cell breaking down that dipeptide or tripeptide before we move into the plasma. So our third topic is going to be the breakdown of lipids. So we're going to think about digesting and absorbing lipids. So let's remember why we group lipids together as one group in the first place. So what's a defining kind of characteristic of lipids? So we're recalling way back to A&P 1 or maybe even to some of your prerequisite courses. Okay, take another couple seconds on this one. Let's take a look. Okay. Yeah. So our lipids have a variety of forms, but what's going to be true about kind of all of them is that they have at least a large nonpolar and hydrophobic region to them. So lipids are a lot of like hydrocarbon chains. Carbon and hydrogen have roughly equivalent electronegativity. Nobody's hogging electrons, so we don't have a charge, which means they don't mix very well with water, right? So if you're making salad dressing, right? You might have trouble mixing that oil together with, say, some vinegar, right? If you pour oil in water, they don't mix together well because of this non-polar hydrophobic quality. The same thing's happening in your gut. Where we saw this word amphipathic before was specifically just for the phospholipids. So they had that additional phosphate group that allowed them to have a portion that can touch water, that polar head. But that was really a modification to the lipid form, which is really that kind of fatty acid tail. It's going to be really characteristic of a lipid. So lipids can be all sorts of shapes, right? We had waxes and fats and eicosanoids and cholesterol, steroids in general, right? So lots of different shapes if we're looking at the molecular structure, but they're all going to have this kind of characteristic of how they interact with their environment. So that's going to be the setup for like why we need to digest lipids a little differently than our carbohydrates and our proteins, both of which have like polar or charged regions that let them mix well with water. Our lipids, because they're nonpolar, because they're hydrophobic, are going to tend to form like, you ever heard that like the pipes have like a fatberg in them and get blocked, right? That can happen in your gut, basically. So we need different strategies for breaking them down. So a typical diet should include about 50 grams per day of lipids. 90% of that is going to be in the form of triglycerides, so like our fats and oils. So the triglyceride is the one that looks kind of like a flag. We've got that glycerol backbone. And then we have our fatty acid tails coming off of it. Okay. Very lazy chemistry drawing, but it's that one. Okay. So we have this problem that if you surround our lipids with water, right? So if you surround a bunch of triglycerides, what they're going to do is they're all going to try to escape the water. And so they're all going to try to just be touching the other lipids, right? We're going to try to limit the surface area touching the water as much as possible. So this is going to make it difficult if we're digesting or absorbing, because you can only really digest or absorb something that touches an enzyme or touches a cell membrane, right? So if we've got them all kind of stuck together, doesn't matter how much we churn our stomach, how much we move our intestines around, we're going to end up with a bunch of lipids that basically the body can't touch. So we're going to form these fat droplets, basically. And in order to digest and absorb a lipid, we're going to have to break those up. Okay. So ultimately, we will have an enzyme that's going to allow us to break these down from a molecular kind of chemical perspective. The word for that will be a lipase. So ase ending means an enzyme. Lip, that beginning, is for the lipid part. These are going to come from the pancreas. But we need to make space for these lipases. We need them to actually be able to attack those bonds in those lipids. They're only going to be able to do that at basically the edges of our big fat droplet. So we've got like a big glob of fat, a bunch of molecules all kind of jammed together coming down. And the lipases are only going to be able to reach the outside, which means that at first, they're really only going to be able to digest to start breaking down the outsides of that fat droplet. That's a problem. That takes too long. So we are going to break apart that fat droplet with our bile salts. So this is one of the important functions of bile, which we know is being made by the liver, potentially stored in the gallbladder. The liver keeps making it. We can store it until we need it, until we eat a fatty meal. Okay. So what's going to happen is we're going to take that bigger fat droplet, and the bile salts are going to let us break it into smaller droplets. And this process is called emulsification. So if you see like emulsifiers in your food or in a food label, that's what it's talking about. It's talking about adding something in that allows the lipid and these aqueous environments to kind of combine. So here's how that works when we're thinking about digestion of lipids. Okay. So our bile salts come from the molecule cholesterol. Right. So cholesterol has this kind of like steroidy form. Right. That's this four-ring stuff. Okay. But we're going to add some stuff so that they are going to be able to mix well with water as well. So that's why they're going to become bile salts. So they're going to have other regions that are more hydrophilic, able to combine. All right. Okay. So we're going to have these bile salts in the bile coming from the liver and the gallbladder. We're going to secrete that bile containing these bile salts into the duodenum, so into that first segment of the small intestine. Okay. So our bile salts, what we're meaning when we say that they have this cholesterol portion, this traditional nonpolar portion that's hydrophobic, that's the part that can touch our fat droplet. The more polar part is the part that's going to touch the water. So what we're saying really is that they're amphipathic, just like our phospholipids were, right? So just like our phospholipids had our polar head and then those fatty acid tails, right? Here we can see this steroid portion looks like cholesterol, like our typical lipid, right? But we've got some places where we have some polarity coming out. When I look for polarity, I just look for basically oxygen atoms kind of sticking around. They tend to produce differences in electronegativity and therefore like dipole moments and stuff. Okay, so we've got some polar regions to this molecule, which means it's amphipathic. It's got both a hydrophobic portion and a hydrophilic portion. So we can see what that looks like here. So we have a hydrophobic side, a hydrophilic side, which means we're going to have a portion that can touch the fat droplet and kind of integrate there. We're going to have a portion that lets water get in near to that hydrophilic polar portion, which is going to help us break stuff up. So that's how our emulsification is working. So we're going to start out with our big fat globule, right? So we got our big fat globule, all our, I'm going to say triglycerides, because that's 90% of your dietary fat, all mixed together, right? You can see how if you were trying to digest with a lipase, right, you'd only be able to get right here, right? But you have a ton of stuff in the middle that you wouldn't be able to reach with your enzymes. So that's the problem we're encountering here. Then we add our bile salts, right? So our bile salts here, we've got our non-polar fatty region. Then we have this polarity over on the other side. So that's the part that's more hydrophilic, can touch water. So we see how that's integrating with our fat globule, turning it into smaller little fat droplets. So we can see this non-polar part, A&P, right? That's the part that's now touching our fat droplet here, okay? And then we can see that portion that's hydrophilic. So I'm just going to write a P, right? That's the part that's out in here, touching that aqueous environment that we find in the GI tract. So as we send these bile salts in, and they kind of wriggle their way in there, which they can do because of that hydrophobic nonpolar region, this has the effect of breaking our fat globule into these smaller droplets. So once we break it up, right, get ourselves some surface area that we can attack, what we're using are our pancreatic lipases. These are the enzyme we need in order to break down the molecules, right? Break down the triglyceride molecules rather than just like separate the globule. Okay, so for our triglycerides, we have this kind of structure. And what the lipases are going to do is they're going to kind of like chop off some of these fatty acid tails. Right, so we're going to be kind of cutting it up. Okay, so we're going to end up with fatty acids, just free kind of on their own. Okay, and we're also going to end up with this kind of T structure. This is a monoglyceride. Okay, so we still have that glycerol stuck up there, and then one fatty acid chain kind of left. Okay, so our lipases start churning up our triglycerides, get us these different parts, okay? Then we can start to actually absorb our fatty acids and these monoglycerides, right? They're non-polar, hydrophobic, so they actually integrate well with your plasma membrane, so you can kind of move them into the cell relatively easily. But they don't all immediately get absorbed into the cell. Some of them are still going to be floating around in the lumen of your gut in the form of what we call micelles. So micelles, we did mention, again, back in A&P 1, what a micelle is like kind of like the small form of these flat globules and fat droplets. We talked about them as a different form that phospholipids can make if you drop them in water. So one thing we can end up with is our phospholipid bilayer. A micelle is where we just have like kind of all the tails of stuff facing in, right? So that's kind of what a micelle would look like. So some of these digested triglycerides in the form of the fatty acids and monoglycerides are still going to be in little collections in these micelle structures. And what's happening as they move through your gut is basically there's an equilibrium between the micelles and the fatty acids and the monoglycerides that are being formed and absorbed. So I think we got a picture of that next. Yeah. Okay. So here's what we're looking at, right? So this is the picture from before with our bile salt. So this is that hydrophobic region, that hydrophilic region. This is our fat droplet. You can see that in our fat droplet, we've got all kinds of lipids, but we've got specifically a lot of triglycerides, right? So when lipase comes in from the pancreas, it's an enzyme. So it's going to break down those triglycerides. So we can see it grabbing on to a triglyceride and catalyzing this reaction, giving us the monoglycerides. So this is what I meant by the kind of T-shaped one. And these free fatty acids. Okay. So some of these are just getting straight up absorbed into the absorptive cell directly. So we can see that happening along this pathway. Right? We don't need a channel. We don't need a transporter. We don't need anything because they're non-polar. So they can just squeeze through those phospholipids of our phospholipid bilayer, this membrane. Some of them are sticking around in the form of micelles. Okay. And they're at equilibrium. So we basically have to kind of control gradients of things, amounts of things, in order to get those micelles to release further fatty acids and monoglycerides for us to absorb. Okay. So in scientific terms, right, more formal terms, what's happening across that apical membrane, right, is that we have simple diffusion specifically. Right, so simple diffusion is the term for that type of transport where we don't have a carrier, we don't have a transporter, we don't have a channel. We're just going directly across that membrane. Simple diffusion has to happen along a favorable gradient because we're not putting in energy, right? So we're going from where there's lots of fat, right? So high concentration of these fats to lower concentrations of fat in the absorptive cell. Okay. Once we have those fats inside the cell, the absorptive cell is a type of epithelial cell. So it's one of our types of cells that's lining our intestinal tract. So it would be part of that like mucosal layer on the inside. Cool thing's going to happen in the cell is we're going to take these fats that we're absorbing and we're going to kind of like modify and repackage them. So this is happening inside the smooth endoplasmic reticulum and then also in the Golgi body. So we take these individual little fatty acids and monoglycerides, and we're going to remake some of our triglycerides. So we had to make them small to get them across the cell membrane, but now we can build them up back bigger again in the cell. Right, so that's one thing that's happening in the smooth endoplasmic reticulum. And the other thing that's happening is in the Golgi body, we are packaging them in with other types of molecules to become something called chylomicrons. So chylomicrons are going to be like combinations of like lipids and proteins, and they're going to be pretty big. So we're not going to be able to let them diffuse across that basolateral membrane. We're going to have to package them all together. So they're packaged into a chylomicron. And then we're going to have to secrete them by exocytosis. So kind of think like how we were getting neurotransmitters out of a neuron into the synapse, right? We have that like vesicle, that synaptic vesicle that then like burst and freed the neurotransmitters. We're getting the chylomicron across the membrane in the same way, right? So we're folding that membrane and letting a chylomicron go out, basically. Okay. And then the chylomicrons, unlike our carbohydrates and proteins, those ones go into plasma. Our chylomicrons actually go into your lymphatic system instead of into the veins, which is cool and weird. Okay, I think we have a picture of this. Yeah, okay, cool. All right, so up here, again, we see the lumen, so the hollow part of the gut up top. So this is our apical membrane. That's that brush border with the microvilli on it, okay? And we can see here those monosaccharides and the free fatty acids. So this is after the lipases have gone in and attacked those fat droplets, broken down the bonds in a triglyceride. So they're moving in one by one into our cell. So here we see them again. In the smooth endoplasmic reticulum, we might just build them back together. Okay, so we might just make a new triglyceride. Right, so all we did was kind of pull that into the cell in that form. Okay, we're going to take those triglycerides. We're going to mix them up with other lipids and even some proteins. Send them to the Golgi. Makes a nice chylomicron. So we've got this kind of lipoprotein structure. It's a combination of lipid and protein. Okay, so in this chylomicron, we can see that merging into that basolateral membrane and going here out of the cell through the exocytosis process. It's skipping over the bloodstream. Okay. And instead of going into the plasma and into the blood, it's going into our lacteal. So the lacteal is like a projection of your lymphatic system. So the lymphatic system is kind of running like parallel to the veins. We have lymphatic veins. They're filled with lymph. In diagrams, they're pretty much always green. So that's a heads up while you're looking at pictures. So we can see that chylomicron going into the lacteal. Okay. And then it's continuing along with all your lymphatic fluids. So we've got fluid that we're returning from that interstitium, that space between the tissues. We know we need to do that because our net filtration pressure is causing like 3 liters of fluid extra to be kind of built up in that space, right? So we remember from our vasculature unit that lymph is part of how we're recapturing that. Right, we have some immune cells here in our lymphatic system as well. Now we've got these lipoproteins, these chylomicrons, circulating around until the lymphatic system joins back up with your cardiovascular system. Your lymph ducts basically dump into your veins eventually. After they dump into your veins, we've got these products in the plasma to be distributed with the blood. So we got a longer process for the absorption of lipids and pretty much mostly fats than we did for our carbohydrates and our proteins. We've got a longer journey here. We've got to go through a couple additional structures before we're ready to send them back to the rest of the body. So here we can see how that integrates a little bit with our liver as well. Right, so our liver is making the bile salts. So that's part of the way it's relating to our lipid digestion. So lipid salts here in our hepatic duct, then going into the common bile duct after the cystic duct from the gallbladder comes in, links into the duodenum. Now we got bile salts here that let us turn our big fat globule into some smaller fat droplets. And here we can see for those bile salts, we don't want to have to remake them, right? Like we can make more. We will make more if we need to. But once we're done with those bile salts, they're just an emulsifier. They didn't like break down themselves. They just separated out that flat globule. So they're just still kind of there in your intestinal tract, passing through your small intestine. So when they get to the end of the small intestine, the ileum, right, because we have duodenum, jejunum, and then ileum. So as we get to the ileum, we can reabsorb those bile salts and pull them back to the liver so that we can kind of reuse them. So here we see those bile salts going into the capillary network that you would find like in the mesentery that's attaching the small intestine to the posterior abdominal wall. Okay. So we're now in the plasma. We got these bile salts in the plasma. And we have this portal system, right? This portal system of veins in the abdomen means that before this blood returns to the heart, it's got to pass through the liver for like filtering basically first. So we can see those bile salts returning through our hepatic portal vein into the liver. So this is where we could recapture them and reuse them if we want to keep reusing them for further digestion of lipids. That brings us to more kind of like niche topics. I don't know. Some other grab bag things that we're absorbing. Okay. So when we talk about vitamins, absorbing vitamins from your food is important, right? It's important to have these vitamins because they often act as like cofactors for various enzymes in your body. We need them to do various metabolic processes. We're not going to go into that in detail, but we need them, right? Okay. So vitamins tend to be either fat soluble or water soluble. So this is linked up a little bit to their chemical properties, right? So think things that are hydrophobic, nonpolar are going to tend to be fat soluble. Things that are hydrophilic are going to tend to be water soluble. So we have like two different classes of vitamins. A lot of them are fat soluble. So here we can see that vitamin A, vitamin D, vitamin E, and vitamin K, aka potassium, are examples of fat soluble vitamins. So these are going to move along the same pathway as the lipids. So if you have a fat soluble vitamin, it's coming into your body and just mixing in with the fats, right? That's what fat soluble means, is it mixes well with the fats. Okay. So since it's mixing in with the fats, it's going to get absorbed along with those lipids. So it's going to dissolve into those lipid droplets. It's getting separated out as those bile salts are attacking that larger fat globule. It's going to end up integrated into the micelles. It's going to diffuse across that apical membrane, and they're going to get packaged into chylomicrons. Okay. So that's how your fat soluble vitamins are getting in. Water soluble vitamins are going to need their own transport proteins to cross the cell membrane. So they're not getting carried along with something else, right? We need to actively have different mechanisms for absorbing our water soluble vitamins. And vitamin B12 is its own kind of like special case. So vitamin B12, we mentioned briefly, at least once before, probably a couple times before. But one of the places that we mentioned vitamin B12 was when we were talking about secretions of the stomach and things that we'd see in like gastric juice. So we need intrinsic factor for the absorption of vitamin B12. So we have to like bind those two things together before we can absorb vitamin B12. Okay. So when we are thinking about those vitamins, they tend to be like larger molecules, right? So we also are directly absorbing some minerals. So sodium is a big one, right? So sodium, our ion, it's getting absorbed. You do need salts in your body the way that that sodium gets absorbed is there's something called solvent drag. Basically, what's happening is as you're reabsorbing or absorbing water, I should say absorbing since we're in the intestine, right? Absorbing because it hasn't been part of your body yet. And the nephron, it was reabsorbing because it was already in your body. Okay. So sodium is going to get dragged along as you absorb water, and then similar to the way it got reabsorbed in the nephron. Then we're going to actively absorb that sodium in the jejunum, ileum, and colon as well. So we're going to kind of be using some energy as well in certain places. Chloride ions, since they're negative and sodium's positive, they just kind of passively follow that sodium absorption. And potassium absorption can be passive as well. Sometimes we will see potassium secretion. So that's kind of like a special case. Usually we're just pulling stuff into the body, past potassium. Sometimes we're getting rid of some of the potassium as well. Okay. Another thing that we want to make sure we can trace is bicarbonate. Okay. So bicarbonate, we know that we were using that in certain places of our GI tract to kind of buffer out acidity, right? So one place we saw it was in like the mucus that's lining the stomach, right? Hydrochloric acid coming out of those gastric pits. It's a pH of 2, right? Super low. We don't want that to mess with the tissue of your stomach. So we had some bicarbonate lining the stomach. Then we also had bicarbonate coming into the small intestine in the duodenum, because the chyme coming out of the stomach is hydrochloric acid, right? That gastric juice mixed in with your food. So we don't want that chyme and that acidity to damage the small intestine either. So we've been adding in bicarbonate with those various secretions coming into the duodenum to neutralize the acid as it passes out of the stomach in the chyme. Okay. So we've been making bicarbonate and putting it into our system. We are also going to want to pull bicarbonate into the body as well, right? We need it in our plasma to buffer out our plasma pH, as we saw, right? That was part of our defense mechanism against pH changes when we were thinking about the renal system and the kidneys, right? And we even saw that some of the reasons we might have disturbances to pH had to do with, like, if you eat way too much baking soda, right, you might end up in metabolic alkalosis because of all that bicarbonate that you've suddenly added to your system. All right, so we know that we must also be absorbing some of the bicarbonate from our diet. Okay, so in the jejunum, right, we've already passed through the duodenum, so we used that buffering function to buffer out acidity. We get to the jejunum. So in the jejunum, any bicarbonate ions that we have traveling through the jejunum, we're going to passively absorb them. When we get to the ileum and the colon, sometimes we are going to start secreting some of our bicarbonate. And the reason we'd be doing it is we'd be secreting them kind of in exchange for some chloride ions. All right. Calcium, we also need as a dietary mineral. So we need calcium to build strong bones, knock out osteoporosis, and we need it in order to help us absorb vitamin D. Those two things are kind of linked. Okay. So for calcium, we're going to be actively absorbing that, again, in the first two parts of our small intestine. So in the duodenum and jejunum, we're actively pulling in calcium. Something that's happening with the calcium is it's binding to a protein in the brush border. So in that apical membrane of the cells, that brush border with the microvilli, you can see why I was checking that we knew what brush border meant at the beginning of class, right? So we have a protein on that brush border. It's called calcium binding protein. It's helping us actively absorb that calcium. So we're transporting that calcium actively into our epithelial cells. And then on the other side of the cell, so on the basolateral membrane, we're going to take that calcium. We're going to run a calcium pump on the basolateral membrane. That's going to take the calcium that we've just absorbed into the cell and pump it out towards the blood into the plasma so that it can circulate around to your heart and then through your systemic circuit so that you can use the calcium where you need it. Right. Iron is a mineral that we talked about when we talked about the life cycle of erythrocytes and the composition of hemoglobin. So one of the places we're getting iron is dietary iron, right? The other place was basically we were kind of recycling iron from old red blood cells. So this is what's going on with our dietary iron. So we know that to move iron around, to transport it, we need transferrin. So trans going across, ferrin, anything ferrous, that's referring to iron, okay? So that was like when we saw the T iron in some of our blood cell diagrams, right? Okay, so the transferrin is secreted into the lumen, so into the hollow part of the small intestine by some of our enterocytes. So those enterocytes are really part of the epithelial lining, the inside of the gut tube, right? Some of them are absorptive cells, some of them are helping us absorb stuff. Other ones are doing other jobs. So the transferrin is being secreted by some of those enterocytes. The transferrin binds to iron, gives us that T iron complex, right? So we have iron and transferrin bound to each other. There are receptors for this T iron complex on your cells. So when this T iron finds an available receptor and binds onto it, this is a signal to the cell to perform endocytosis, which is just the opposite of exocytosis, right? So you're taking like a chunk of the membrane and just pulling it all in. So we're pulling in the T iron through endocytosis when it binds to this receptor. Okay. Once we have that iron in our bodies, some of it we're going to store as ferritin. So this is like when we saw F iron, right? Some of it we're just keeping. Some of it we're going to keep just right in those enterocytes that we pulled it into. Some of that iron is going to go into your blood, into your bloodstream, travel around. Particularly, we saw one of the places it's traveling is to the liver when we were talking about the life cycle of red blood cells. So in order to transport that iron around in your blood, you'd need to bind it to transferrin again. So you'd end up with that T iron complex across the basolateral membrane, going to your bloodstream into the plasma, going into a vein that's going to filter into your liver. Your liver kind of mediates stuff, adjusts stuff. And then if we're building new red blood cells, your liver could release it so that you can make that heme group for your hemoglobin. Okay. So we'll pick up with water next time as a good pause point.